Yeah, I might switch now from phenotyping or rapid generation cycling back to a bit more of like statistical genetics with a bit touch of genomics maybe, by introducing the concept of haplotypes and ways how to identify and how to track haplotypes in breeding material. And of course, that there are many different ways to think about haplotypes and many different resources that you could use to actually perform haplotype analyses. But I want to talk about them like specifically in the case, or in the context of genotyping data generated from very popular, very common high throughput genotyping platforms as we know them in wheat. For example, SNP genotyping arrays or genotyping by sequencing platforms. So yeah, the idea would be that after this small lecture, you would just have like a few more ideas what you could actually do with the data you probably already have at home, at your home institution, and maybe a few ideas how to play around with this. And I hope you find this any helpful. So I've structured my lecture into two parts. So first of all, I'm going to talk about QTL-based haplotype analyses. So what you can actually do as a subsequent step after you've identified a genetic signal, after you've mapped the QTL. And in the second part, which is actually the one I'm a bit more looking forward to, I'm going to talk about like a newly developed approach that we've been using recently that aims at the identification of genome-wide haplotypes. And I'm going to introduce the concept of local genomic estimated breeding values. So to track down chromosomal fragments with a high variance in terms of phenotypic values. So speaking of genetic mapping approaches, maybe just to get everyone on the same page. So obviously there are many different popular approaches. QTL mapping was a really big thing 1 or 2 decades ago, especially. We have a few properties, a few limitations here. So we have a fairly low mapping resolution with QTL mapping. The number of alleles, if we consider single chromosomal chunks or blocks, is relatively low. We only have 2 alleles because this all goes back to the parents. Statistical power, on the other hand, is pretty high because we normally have pretty balanced frequencies of alleles. But another downside is that the population type is always really experimental. So you make a cross, a biparental cross, and then you have a population. And in most cases, once you've identified a QTL, you would never find it again in another population. So that's why it's always highly experimental. Another approach that originated from human genetics, which has been like widely used in plant and wheat genetics as well, is genome-wide association studies, or GWAS. Compared to QTL mapping, you have like a way more enhanced mapping resolution, because you're actually not using a few recombination events that you find in your biparental population, but you're actually tracking recombination events that can go hundreds and thousands of years back. That's why the number of alleles, if you consider chromosomal blocks normally, is also increased drastically. On the other hand, the statistical power to detect variants in the genome that actually do have an effect on your trait of interest is comparatively low. Normally in wheat genetics or wheat breeding research, we're normally working with fairly low numbers of genotypes in our population. So considering the rare allele, or the model that says that rare alleles tend to have a high impact on a phenotypic trait, it's sometimes really hard to track those rare molecular variants. But the advantage of GWAS is that we can basically assemble our population just the way we like. It's highly flexible. We can just include or exclude genotypes. So that's definitely an advantage. And last but not least, we have joint linkage and association mapping. I'm now speaking of nested association mapping populations that have become really popular because they're trying to combine both positive features of QTL mapping and GWAS. So here we have a fairly high mapping resolution. Number of alleles per chromosomal block are fairly high. we have a pretty high statistical power compared to GWAS because we have kind of a nicely structured population, and even rarer alleles are sufficiently represented. Probably the biggest disadvantage is that the population type is still pretty experimental, so you have to create an M population at first, which can be like a really time-consuming and costly process. With technologies like speed breeding, it's way easier, but this might not work for winter crops, for example, winter wheat. So yeah, the most popular approaches probably are GWAS and joint linkage and association mapping approaches, and I don't want to go into detail, but from my experience, I feel like mixed models are most commonly used when it comes to mapping genetic signals, in which marker effects are modeled as random, just like Ben was introducing this morning. Typically, these mixed linear models include population structure, by fitting principal components or Q values as a fixed effect in a model. This is something that is really commonly done, and there's a whole bunch of softwares or R packages that you can actually use. For example, GeneAble, which originally comes from human genetics, but which is a really nice and powerful tool. Gapit is another R package. There are standalone softwares like Tassel or Plink. Plink especially becomes really useful when you have to handle like really large data sets. But yeah, there's a whole bunch of other softwares. Is anyone in this room using anything else but these tools, or are there any other packages that you think are useful? Or has anyone ever used Tassel, for example? Yeah, probably the American people here in this room. Anyway, so there's a whole bunch of things, and it's I don't want to go into detail on that. But talking about genetic mapping or QTL mapping or genomic signal detection approaches, I want to briefly summarize the high throughput genotyping platforms there are in wheat. And this is actually not a table of mine. This is modified from a publication from Bassi et al. It's already 2 years ago, so the numbers might not be completely up to date, but we see different platforms, starting, for example, with CAST markers on the top left, where you usually have fairly low cost per sample, but you can only consider a low number of markers at a time. You have different Illumina SNP genotyping arrays, the classical 90K array that has been like really commonly used, widely used, the improved version of it, the 15K array. We have different genotyping by sequencing platforms, mostly provided by universities. We have dart markers, which I haven't been using so much until I moved to Australia. Australians love dart markers. Well, yeah, they're pretty cheap. Probably that's the reason. We should probably tell Germans how cheap they are, so they would use them too. But yeah, so this is basically more or less like very popular genotyping platforms you can find in wheat. And what we see here is that SNP markers are really predominant across all these different technologies, which is fair enough because SNPs are basically everywhere. They are really abundant. That's why they're also pretty cheap to score. So they're basically the ideal marker type to use. But the downside is SNPs also have a few limitations. For example, they're typically biallelic. So you only have like a limited informational content when you wanna consider chromosomal blocks. So you always only have allele A or allele B. And that's why you can also run into limitations when you want to depict a SNP trait association, because you basically can just say, okay, this allelic group has this phenotype and that allelic group has this phenotype. So it's not really high resolution. And one possibility, and of course like now I'm getting there finally, is to look at haplotypes. And I've used this word an awful lot, like probably 100 times already in the first couple of minutes. But what is a haplotype actually? Is there anyone who might give it a try? Like, what is a haplotype? Have you ever heard of a haplotype? Does anyone give it a try, like in terms of definition? Oh, probably not. Okay, it's a bit awkward. Well, I like to refer to this statement an awful lot. A haplotype, that's just like 2 or more SNP alleles that tend to be inherited as a unit. And it's a really straightforward definition, really clear, and I like to refer to this a lot. Also because Rex Bernardo said it in one of his books, and Rex Bernardo is pretty famous, and it's always good to refer to stuff that famous people said. So that's what I mean by referring to a haplotype. It's two or more alleles that tend to be inherited as a unit. And if you try to think about this graphically, I really like to refer to this recent publication by Bevan et al. A lot. So this is how you can think of a chromosomal block here, where you have 4 different genes lined up, and each of the dashed lines would represent a single nucleotide polymorphism, a SNP basically. Of course, in wheat we don't go nowhere near like this in terms of coverage of markers. But let's imagine like we had such a situation. So we have intergenic markers, intragenic markers covering the exon and intron space. And when we think of haplotypes, we're not really interested in. We're not really interested in each of these single markers separately, but we would just group them together to form a unit, the one that Rex Bernardo is referring to. So we would really consider a whole bunch of markers, almost 30 in this example, as one single unit. And to go further, we're not even interested in including all the markers. We're really not interested in those markers that give us monomorphic signals across our genotypes. What we're really interested in is only tag SNPs, so-called tag SNPs that produce polymorphisms in the population we're looking at. So what we're effectively doing is we're reducing the complexity of looking at 30 markers. We're reducing this whole thing down to a block, to a haplotype block, and look at variation for this particular block. In this example, for example, here, you see 3 different haplotypes when you consider 30 markers. So we've drastically reduced the complexity, which is pretty good. Okay, so that's the basic definition of a haplotype. But how do we get there if we apply classic QTL detection methods? So how do we get from QTL to a haplotype? So I'm sure you're very familiar with this example here. So I'm mostly working in hexaploid wheat. We see the 3 different genomes in nice different colors with 7 chromosomes each. And if you're really lucky and your phenotypes are good and your population's big enough, you might map something like this, a really nice peak on a chromosome. In this example, it's 7A. And you say, okay, I've mapped a really nice QTL. What many people do, from what I can tell from publications, they would consider those single markers alone. Try to, for example, group the population based on the most significant marker, look at the phenotypic distribution across the different allelic classes that this single marker gives you, which is essentially only 2 in the case of wheat. But what if we just consider this whole chromosomal block, all of the markers at once, and maybe even the markers that are close to this QTL? So here you see essentially the same picture. All I did was just to zoom in. So we have the same QTL peak here on this chromosome. It's a zoomed in photo. Actually here you're seeing a QTL for the same trait scored in 4 different environments. Doesn't really matter. It's a pretty stable QTL. And it's actually pretty interesting to look at the LD structure in this block. So in this example over here we had like 5 very, very significant markers. And if we zoom into that region and if we look at linkage disequilibrium, as you see down here, we would see that those markers are actually very, very strongly correlated, or the marker alleles are very highly correlated. They form an almost perfect LD block. So what you can then, and that's actually very critical. So this whole marker assignment to haplotype blocks works most efficiently on the basis of linkage disequilibrium. So what you could just do is, going from there, you could just take the 5 different markers over here, 1 to 5, and line up the different possible allelic combinations that you find in your population of interest. And again, that's super flexible. It's really flexible. You just kick out a whole bunch of genotypes because you're not interested in them. Might give you a different picture, but that's really it's a really easy and straightforward approach. So in this case, considering those 5 markers from the QTL peak, that forms a perfect LD block. we find 5 different haplotypes for these markers, with the first 2 being the most abundant ones in the population, almost split 50-50, and the other 3 haplotypes being less frequent, if you will. Oh, sorry.
When you talking about the haplotype block based on the LD, they may change. What is the criteria to select?
That's a very sorry, the significant markers, or
no? What is the criteria to develop a? Because we are also working on this on some of the pre-breeding stuff we have developed. So we have used some early criteria. I'm just wondering.
Yeah, no,
what is the?
That's a very good question. So actually, there is no rule. There is no general rule. So I would say if your population is fairly small, I would be very strict in terms of LD, because population structure is one of the driving forces when it comes to LD. So if you have a small population, say 150 genotypes only or so,
We have
I'm sorry.
We have thousands.
Oh, thousands, yeah. Well, then you could probably, I would consider an LD of 0.5, 0.6 if you have a few thousand genotypes. I would say that's pretty high. Yeah. Whereas in my smaller populations, I don't have. I'm not fortunately enough to have such big populations. I would increase the threshold, to be honest. Yeah. All right. So these are the different variants we would find for this haplotype block on this chromosome. And what we can then do here, which it's probably not so extremely relevant for a practical breeder, but speaking from an academic point of view, you can make an awesome figure based on this analysis, which is pretty important if you want to publish your results in a journal. So we could actually, based on this information, we could really easily calculate something like a haplotype network. And let me just briefly explain you what you see. So each of those nodes represents one of the different haplotypes. And what the network basically tells you, or it calculates the genealogies between the haplotype sequences. We're now treating the 5 markers as a sequence. So it basically tells you, in theory, how many mutations would you have to get from one sequence to the other. And yeah, you can. There's a whole bunch of free packages out there, like PopArt, for example, which is a standalone software, or Pegas, which is an R package. A whole bunch of softwares that allow you to construct such haplotype networks, and they also very nicely graphically visualize how genetically or how distant basically different haplotypes are. For example, in our case, a haplotype 1 or 2. So, and in the last step, you could then try to translate this haplotype group assignment to actual phenotypes, to a trait of interest. Could be anything. Could be a resistance. Yield is always a bit tricky because yield is so extremely quantitative. But you could basically do this for any trait you want. So here we're just grouping our genotypes based on the haplotype assignment, based on the haplotype they actually carry, and we compare their phenotypes. And what we see in this example here is that haplotype 1 is always, the genotypes in the haplotype 1 class always have the lowest phenotype, and that there are different other haplotype groups that actually have a significantly higher phenotype. So we're now trying to increase the resolution of the marker trait association, if you will. So I'm now gonna show you a few examples from actual data. First example is from Samir. So this guy here with a shovel. He's sitting somewhere in the back. He was nice enough to provide some of his data that he's going to present in detail on Sunday afternoon. It's an awesome data set. He's working on durum wheat and drought and crown rot resistance. And he was crazy enough to actually work on wheat roots, which is one of the most frustrating things you can do, because they're really hard to measure. You can see him there, like in the field with a shovel, so it's really hard work. But he also did some phenotyping in the glasshouse, and luckily Lee already covered this. So he phenotyped a whole bunch of genotypes for root angle, so the angle at which roots disappear a few days after sowing. And then luckily he was rewarded by the god of statistical genetics with a really nice peak, if you will. Now seriously, that's sometimes really frustrating. So sometimes you're not that lucky, but he was. So he mapped something super significant on chromosome 6A. And just following the general procedure that I've just described, he was looking at LD, and he decided, oh well, I'm gonna consider all the 7 markers that are in pretty strong LD with each other for a haplotype analysis. And this is what he does. So this is what he did. So he constructed a haplotype network using PopArt, which is one of the softwares I've just mentioned before, and he found 8 or 9 different major haplotype variants, with hap1 and 2 being the most prominent one, and you find a super significant difference of 8 degrees in root angle between groups 1 and 2. But as you see, there's a whole bunch of other groups. Actually, the color indicates the phenotype. There's a whole bunch of other groups with similar phenotypes, which is pretty cool. So Samir then went on and compared phenotypic measurements from the field, actual field data, between, say the main 2 haplotype groups, as you can see here. So on the top left, you see yield under drought conditions, and drought. We have a lot of drought in Australia. So what you actually can see here is that there is a significant difference in yield or in field performance between those 2 haplotype groups, which is pretty amazing, because remember, we group these genotypes based on their allelic state of a region in the genome that has been found associated with root angle. So that's actually pretty awesome, because this trait, after a few steps, translate directly to performance of genotypes in the field. And the same was true for a stay green trait. And Samir will talk a lot about this in detail on Sunday. What I'm just trying to show you is that it's actually pretty cool to translate a peak that you find for a trait like root angle all the way to performance in the field. So another example comes from a study that I was working on a couple of years ago, where we measured root biomass, which is probably even worse because you have to dig them out and you have to dry them and you have to weigh them. But I was also fortunate enough to map 2 significant QTL on chromosome 5B. And so we basically did the same thing. We considered both of these QTL peaks, looked at LD in that region, and we constructed haplotype networks just in the same manner as I was explaining before. The only difference to these networks here is that we didn't color code the nodes according to the phenotypic measurements, but we color coded the nodes according to the origin of the genotypes that are in these different haplotype groups. And really excitingly, what we found was that there are 2 haplotype alleles only present in Chinese land races, which are pretty distant from the most common European material. And whenever these two very rare variants are present in a genotype, you get somewhat like an explosion in root dry mass. So this specific combination of alleles is actually associated with a dramatic increase in root biomass. So this was pretty exciting, and we're still doing follow-up work on this. So to sum up the first little bit. So the general idea is to reduce the complexity on the one hand, but to increase the resolution at which you can actually depict marker trait associations. And obviously the main goal would be to stack as many favorable haplotypes as possible. So going back to the publication from Bevan et al. That illustrates how to group single SNP markers into haplotype blocks, we could now think a bit further. So all these 30 markers here, they basically now form a new locus, something like a marker, if you will, that just occurs in different allelic states. So we increase the allelic diversity of this locus, and what you can now think of as a breeder is just stack as many favorable haplotypes as possible. You can think of different traits, and it's down here in the bottom. I hope you can see it. So you could think of locus 1, which is this guy here. You want to select for the latest firing time, but at the same time, obviously, you want to select for the highest yield, which is somewhat associated to molecular variation at locus 4. You do the same for disease, the same for protein, and so on. So the general idea is really to stack up as many positive haplotypes as possible. So to formally define a workflow, how you get from QTL to haplotypes, if you just want to play around with this using your own data at home. So you would firstly need a genomic signal for your trait of interest. That's essential, obviously, in the very beginning. You could, for example, run a GWAS, map a QTL. Then you investigate local LD in that block. Because in wheat, it's almost always the case that if you're looking at a trait that is important, that this trait has been on a pretty strong directional selection, and that there's actually something like an LD block somewhere in that region. So that's really worthwhile. So based on linkage disequilibrium, you can then define haplotype blocks. And again, your question was really good. There's really no common rule, no common ground for this. So you would really have to try and error, basically. Have to see what works, and define haplotypes. And then in the next step, you would investigate the actual haplotype or the sequence diversity in your population of interest. So obviously, in the worst case scenario, if you had, say, 10 SNP markers, worst case scenario would be that you had 2 to the power of 10 different possible arrangements of alleles. But this is almost never the case, never. So normally you end up with, say, 3 or 4 or 5 variants when you investigate the sequence diversity or the haplotype diversity in your population. Yeah, and last but not least, and that's pretty cool for publications, for example, you can compute haplotype networks and compare different properties of haplotype groups. For example, you can compare the geographic origins, traits of interest, and so on.
Yeah, I think because I'm curious when we are talking about the haplotype network. So I think that is basically based on the chromosome and their position, right? Because we have constructed the haplotype map for each chromosome in B, then try to correlate. So I just wondering when you are saying networking of the haplotype, that is based on the LD between the haplotype and construct.
That's only based on LD. Okay. No information about positions or something. That's literally just.
In our case, we have the information available.
Well, yeah.
On the map.
Yeah, that's interesting. All right. Good. So that was the first bit of my talk today. So in the second part, I wanna talk about genome-wide haplotype analyses, or a pretty easy and ready-to-use approach, and talk a bit about the concept of local genomic estimated breeding values of chromosomal blocks. And yeah, as a German, I brought a case study from winter wheat. I'm glad to see a few winter wheat people here too. So what I always like to refer to as an introduction to this study is this slide here. So here you see the development of winter wheat yields in Western Europe, and it's just impressive. So you see the production year on the x-axis and the yields in decitons per hectare on the y-axis. You could just delete the 0 and say tons per hectare if you're not familiar with decitons. And the data comes from FAO Stat. So this is just something I downloaded, and there are a few things going on. So first of all, compared to the early 60s, yields of winter wheat have increased 2.5-fold, which is just, it's pretty crazy, pretty massive. So farmers are on average now harvesting 2.5-fold as much compared to 50, 60 years ago. But at the same time, we also see a kind of a plateauing trend in recent years, and that's nothing just that's nothing specific to Europe, but this trend you most likely find in any big wheat producing region in the world. And based on this, we were asking a couple of questions. So first of all, we were asking, how high is the actual rate of genetic gain? Because you could always argue that this massive improvement in yields is mostly due to improvement of agricultural practices, that farmers now have access to way improved plant protection agents, way better chemical fertilizers, and so on. So we were really interested in answering this question of how high the actual rate of genetic gain is. Secondly, if there is any improvement in genetic gain, and spoiler alert, yeah, well, there is, which are the actual genomic regions that can be associated with this improvement or with this drastic improvement in yields. And last but not least, the by far most difficult question, and again, spoiler alert, I'm not going to answer this. How far can we go? I think nobody can really answer this. That's way too complex. But I'm just going to show you a really like rough estimate that we did based on the data set that we have at hand. Modern wheat varieties, you can probably exchange wheat by any crop species. Modern wheat varieties only perform well under very high chemical inputs. This is a statement that many people outside our field would absolutely agree with. And I've talked to many farmers that actually do believe that modern varieties, they are good if you add heaps of fertilizers and plant protection. But the old ones are still way more resilient when it comes to unfavorable environmental conditions. This is really, that's reality. This is what many farmers, at least in Europe, think. But obviously, as plant geneticists or plant breeding researchers, we weren't quite convinced. So we were asking how high is the rate of genetic gain. And to answer this, we were setting up a really huge collaborative project in Germany with 8 or 9 different public partners, in which we put together a collection of almost 200 registered wheat varieties. And it's really important to note here that this is not a classical diversity panel, randomly sampled lines, but all of these lines played a really predominant role played a predominant economic and agronomic role by the time they were released. So these are really like the top performers of each of the different years of registration. And we tested these lines really extensively in large field trials across Germany, covering a whole range of different environmental conditions. And the coolest part about this project is that we didn't only test the genotypes under optimum conditions, where we added all loads of fertilizers and plant protection, but we tested them at 3 different management levels, where we stepwisely decreased the management intensity level. In the medium environment, we added fertilizers but no plant protection, so here we had biotic stress kicking in. And in the extensive version, we didn't apply any fertilizers or any plant protection. We looked at 14 to 15 different traits in total. This was a massive project. So yield with all its component quality traits, resistances. And as for genomic data, we had SNP data from the 15K array at hand. So talking about genetic gain, here I'm showing you yield across years and years of registration and locations. And here you see yields for the management intensity level where the plants had or where we applied all or maximum amounts of fertilizer, plant protection. You see a nice linear trend. If we try to quantify it, there's around 15% difference in yield performance between the oldest and the most recent varieties, which is already pretty good. However, this difference between the oldest and the most recent varieties becomes even more distinct if we look at the medium intensity level, where we applied fertilizers but no plant protection. So here we had really strong rust pressures, especially rust, which is a problem in Germany too. And the same trend was true for the intensity level, where we didn't apply any fertilizers or any plant protection. So yeah, going back to those farmers again that say that old varieties still perform better under unfavorable conditions, we can really say, yeah, it's true. Modern varieties, they do outperform old ones under optimum conditions, but they are particularly good when it comes to really stress conditions in the form of nutrient deficiency or biotic stresses. So now talking about identifying genetic factors that underlie this improvement in genetic, or this genetic improvement, we could of course go back and use any kind of mapping approach to map a genomic signal. But each of these approaches here that I mentioned in the very beginning, they have like strong limitations. For example, QTL mapping, which is expensive, not transferable. GWAS, especially in our small population, we would really have serious problems with statistical power to detect anything. And yeah, obviously, joint linkage and association mapping is pretty inflexible and pretty expensive and takes quite a while to set up. And most importantly, all of these classical mapping approaches, they really rely on arbitrary significance cutoffs. So, and Ben was briefly getting into this. So you're basically just focusing on single markers that exceed arbitrarily set significant threshold. And even if you calculate something like Bonferroni or FDR, it's still somewhat arbitrary. And you would basically ignore all the other loci that which we've learned this morning are actually very important for most of the complex traits we're looking at. So we were coming up with a really straightforward and easy to apply approach in which we're using the genomic prediction framework to actually identify important genomic regions in the form of genomic blocks. And the general workflow looks like this. So we would just predict single marker effects, just in the way we've always been doing it, using the classical SNP-BLUP approach. But instead of considering single markers alone, we would group them based on linkage disequilibrium and come up with something called a local genomic estimated breeding value, a local GBV for whole blocks of chromosomes that have been defined on the basis of linkage disequilibrium of the markers in these blocks. And finally, we could, to really pinpoint to those regions in the genome that do have, or that do make a difference in the population we're looking at, we calculate variances for these chromosomal blocks, for these local chromosomal blocks. So we could think of a scenario where we just have a handful of chromosomes. It's not near as bad as wheat, so we only have 5 chromosomes, and we could just focus on one for this example. So we have one chromosome with 30 markers and a length of around 50 centimorgans. So really, really simple. So in the first step, what we do is we predict SNP effects using the mixed model framework that Ben was explaining this morning. So we end up with many, many small effects that are pretty close to 0. So it's probably pointless to pick out any of those because they're all really heavily shrunken towards 0. So the novel concept here is really that we're now trying to integrate this into previously defined blocks. And there are different ways to define blocks. For example, we could do it based on distance. So we could say we split up our chromosome into blocks that are 5 centimorgan of length each, for example. So based on distance. We could do it based on marker numbers. We could, for example, form blocks that are 3 markers long. Option B. The problem really with these two approaches in wheat is that the genome is extremely big, and normally we see huge gaps in the genome where we don't have any marker. So using either of these two approaches, you really you run into the risk of grouping markers that are physically light years apart of each other. So that's really critical. So that's why I'm just going to be focusing on linkage disequilibrium based grouping, because I think that's actually the most elegant way to group markers into blocks. And here you simply do it on the previously defined LD threshold, going back to our discussion. In this case, for example, I set a threshold of 0.7 as our population was not big enough. And then what you do, if you consider different blocks. So for example, this is a very, very big block here, consists of several markers in this example. We now investigate the different haplotypes there are in the population for this specific block. So here we have, in this example, we would have 6 haplotypes for this block. by just summing up the markers, we could get something like a local genomic estimated breeding value for each of those blocks here. For each of those haplotypes here. And using these values, we could simply calculate a variance. And in the last step, we could simply plot the variance in a Manhattan plot, just as we did using p-values from a genome-wide association scan. We could simply plot variances of the previously identified blocks. So that's basically the whole concept. So looking at some real data, so this is data from the experiment I've just introduced. So here we're looking at yield under optimal conditions, and if we predict marker effects for obviously we did it genome-wide, but here in this example we're doing it. I'm just showing chromosome 1. So we see many markers with effects closely around 0. The most or the strongest predicted effects are in the magnitude of plus to minus 2 kilos per hectare. So they're really tiny. It's really hard to based on this pick out the marker that actually has pick out a marker and saying that this marker has a particularly higher effect than another. But at the same time, working with wheat, we know that there are very strong and pronounced LD structures. So we know that there are really tight blocks of LD, and this is not only true for wheat, but pretty much for any crop that has been under very strong selection. And so we know that blocks of these markers, or to be more precise, alleles of these markers in these blocks, they tend to be jointly inherited. So what we did in the first step was we used an algorithm to generate a genome-wide LD-based haplotype map. So we basically just grouped all the markers across the genome that exceeded a threshold of 0.7 into blocks, and we just those markers that didn't exceed the threshold were just we just kept them as single markers. In the second step, we summed up allele effects in the blocks, just as I was showing you in the cartoons in the scheme before. And in the last step, we were identifying those blocks with the highest variances on the trait of interest. In this example, it's yield under optimal conditions. So that's basically the outcome of all this computation. So here we see again the 3 different subgenomes of hexaploid wheat, the A, B, and D subgenome with 7 chromosomes each, and each of the dot represents a defined LD block, if you will. And on the y-axis, you see variance for yield. So it's not a p-value anymore, but now it's variance. And for yield under optimum conditions, we see... it's moving, great. So we actually see that there's a block on chromosome 2D that consists of more than 100 markers. So it's a massively conserved block that spans a region of 37 megabases. And maybe just very quickly to this really rough comparison between megabases and centimorgans in the beginning. I think this block initially spanned 17 centimorgan, but if you look at physical distances, it's actually close to 40 megabases. So it's a massive block. And if we compare yields of all the 191 wheat varieties with the actual predicted effect of this block that they're carrying, we get a picture like this. So on the x-axis you see yield in decitons per hectare, and on the y-axis you see the different estimated block effects for this massive block here. So we have one allele, one haplotype of this block that has a predicted effect of around 0, whereas we have a handful of alleles that have a strongly negative effect, and basically one other allele that has a very strong positive effect. And unfortunately, but this was kind of expected, I guess, most of the very high yielding wheats that we have in our population, so some of them are even exceeding 10 tons per hectare in good years, so they are pretty much fixed for the positive allele of this block. But there's a handful of other lines. This, for example, it's a B wheat, which has moderate to low protein content. So it's a high yielding variety, has a high yield already, and it's pretty recent from 2012 or 2013. So we could think of just exchanging the allelic state of this chromosomal block by just using a handful of tag SNPs could actually have a pretty massive effect in the field, which around plus 30 kilos on average by just changing the allelic state of one locus. So we could now also think of correlations of haplotype effects, for example. So this is what you see over here. So obviously we can predict haplotype effects for the most optimum condition for the most optimal environments, for the medium intensity level, or for the low intensity level. And this is what we did here. And generally, what we see is that we have a pretty nice. Oh, this thing is moving. We see a pretty nice linear shape, actually, meaning that haplotypes that have a positive effect on yield under optimum conditions tend to have a positive effect on yield also under bad conditions, which is pretty amazing. I mean, there's a handful of haplotypes, and if you're really interested, you could always go and look into them in detail. For example, those guys over here that tend to have a zero effect in one environment, but maybe a strongly positive effect in another environment. So to make it even more complex, we could even think of comparing different traits, right? So in wheat, it's not only yield that is important, but also protein content, for example, or resistance. Just got to stop this, otherwise you're getting too distracted. So here, for example, so the color coding is the same. This is the big 2D block, most or the highest positive effect on yield under optimum conditions, the highest negative effect under optimum conditions. And if we plot this against crude protein, for example, a very important trait for quality, we actually unfortunately see that this block with the most negative effect on yield has the highest positive effect on protein content. So here we see like this negative correlation kicking in between yield and quality, which is probably not so surprising, but it's pretty nice. Can't really see a very clear pattern if we try to combine rust resistance, for example. But I'm just trying to show you a way how you can graphically display your data and look at it from different angles. Oh yeah, but most important take-home message is that it looks like that really similar genes or similar genetic factors are actually involved under optimum and stress conditions when we're looking at yield. So I'm getting close to the end. Another comparison we made was we looked at yield trends of all the lines, basically just as I showed you in the very beginning. So you see on the x-axis you see the registration year, on the y-axis you see the yields, and the three colors indicate the three different management intensity levels, and we see really nice correlations between yield and registration year. But if we now consider the 100 linkage disequilibrium blocks with the highest estimated variances on our trait of interest, and if we count the number of alleles that have an estimated effect of 0 or are even negative, then we get an almost perfect mirror image of the yield increase trends. So let me just briefly explain you what you see here. In these plots here, you see the number of so-called detrimental alleles for the 100 blocks with the highest variances. So the count of alleles that either had a negative or a 0 effect on yield. So what we can nicely see here, we can really track this on the molecular level, is that breeders over time basically got rid of detrimental alleles. Because, as you can see here, the most recent lines, they have significantly lower numbers of alleles with 0 or negative effects. And that's also like a very nice feature. So you see some outliers here in each of the plots that have like a significantly lower yield than all the others in this class. And those are the lines that also have significantly higher numbers of detrimental alleles. So you can find this in every intensity level. which is pretty awesome that you really get this mirror image. So last but not least, how far can we go? And I'm gonna keep this one pretty short. So I just played around with the data a bit, and I was picking out those varieties with the highest actual yields across years and environments. So the top 20 varieties that were ranging somewhere between 85 and 90 decitons per hectare. And we were asking the question, what happens if we pick out, say, the top 20 haplotypes with the highest variance and replace the bad alleles with the most optimal alleles, with the most optimal haplotypes, what we did here. So considering the top 20 blocks, we were getting something like a yield increase of 1.2%. Could increase this to 2.6% if we were considering the top 50 blocks. Increase this a bit further when we were considering the top 100 blocks. And in the most unrealistic scenario in which we were to stack all possible positive haplotypes across the genome, we were getting something like an increase of 23% in yield, which is obviously it's overly optimistic. We're ignoring any sort of interactions between genes. We're just assuming full additivity, which is really unrealistic. But I think it nicely shows that even in a very, very narrow gene pool, like the one of European elite winter wheat, there's still some considerable room for improvement if we only were to come up with technologies that allowed us to stack the best alleles in the most optimal way. Okay, so to very briefly sum up, single SNPs are awesome because they're cheap and they're abundant, but they have limited informational content. And one way of getting around this is really to look at haplotypes. And that's actually, the methods I've shown you are pretty handy and pretty cool, I would say, because they are really flexible. So you could easily modify the populations you're looking at. They are very, very easy to use, and they're ready to use. So you could probably go back home and just give this a try using your data sets. Using haplotype analysis, you can actually track origins and trait effects of different haplotype groups, which might be interesting. In our project or in our study, I think we can nicely show that genome-wide haplotype scans are really useful to track down or to pinpoint those blocks in the genome that actually do have an effect. And you can think of a breeding population in your breeding program. So we think this is a pretty cool, pretty easy to use method. And yeah, obviously the main future goal of everybody is to stack as many good haplotypes as possible. And yeah, Lee was talking about speed breeding a lot. Genome editing might be an option. That's probably like the most difficult most difficult bit. Yeah, with this I'm pretty much done. Obviously this was a big team effort. Many different people involved. Most of the partners from Germany, but also my new colleagues from the University of Queensland in Australia. And yeah, I'm really happy to take any questions, if you might have any. If we had time.
Since I'm chair.
Yeah.
Let's just thank Lee really quickly.